someone to speak at my symposium for ASPB 2 years ago, and I talked to Deb Domer, who was a colleague of mine, and I said, I'm looking for a really good speaker who can talk on sort of sustainability and the future, where we're going, and she said, this guy. So I invited him, not ever having heard him before, and he was fantastic. And so I said, I want people at Berkeley to hear this guy too. So that's why he's here today. He's a professor of nutrition at Penn State, but he's a local guy. Actually, he is from Watsonville. And he came here for undergraduate school, and then he went to another school that's, where is that now? No, he went to UC Davis to get his later degrees. And I just, so he and I were going back and forth. I said, oh, I don't know how many people are going to come because we have a lot of seminars going on. And so he wrote back, and this is what he said. When I said, oh, I don't know. He said, if I can encourage one student to think about global food security in their scientific work, my visit will have been a success in my view. So this is your charge. You take account of everybody who's in here, and one of you must go out of here being interested in global food security. So he's going to talk, his title is up here, Roots of the Second Green Revolution, and I invite you all to enjoy this.
Thank you very much, Peggy. Good afternoon, everybody. Thank you very much for coming. It's a great honor to be here, since this, as Peggy said, is my alma mater, and I had so many fond memories and inspirational teachers while a student here. And actually, Lou Feldman is here today, first root biologist I ever met. Norm Terry, who's not here today, is traveling, but my nutritionist, very influential in my thinking about things. And Don Kaplan, who's no longer with us, but some of you older faculty might remember Don, just an amazing instructor who plant morphology. And so I think you'll see today that all three of those threads are things I'm still kind of working on, things that I was inspired to do by professors here at Berkeley. So as Peggy said, this is the title of my presentation today, Second Green Revolution. So what we have is, in the 1960s, yield actually declined when we fertilized plants, things like rice and wheat. They would get tall and then fall over. It's called lodging. And so there was a lot of concern. The population was growing. People were very concerned about mass starvation. Norm Borlaug and other breeders developed dwarf varieties of rice and wheat that you could fertilize and they wouldn't fall over because they never got very tall. And that basically was the basis of the historical Green Revolution that Norm got the Nobel Prize for. Norm passed away several years ago. What are we talking about with you today is the possibility of doing what we see here on the red line, which is that we have good yield at high fertility and superior yield under low fertility. So this then is the area of benefit we've seen in the historical 20th century Green Revolution. And this is what we're talking about needing for the 21st century. Do we need a second Green Revolution? Well, this may be old news to some of you, but just to refresh some of the recent statistics, 850 million people suffering chronic hunger. That number is expanding in Africa. Over 3 million children dying of malnutrition every year. In fact, half of all childhood deaths in the developing world are directly or indirectly related to malnutrition. Of course, the human population is expanding. Virtually all of that expansion is expected to occur in food insecure regions. So people who work in food security are expecting this problem to get worse. And low input agriculture is what's normally done in these places, and in fact it's the single largest human occupation. Low input agriculture is agriculture without the benefit of fertilizers, irrigation, pesticides. And so low input agriculture is low output agriculture. We tend to have very low yields in these environments. Let's take a look at some yields. These are three of the principal world food crops. Maize, the most important crop on earth. On the top bar we see yield potential under ideal conditions, and then the other bars are actual yields obtained in different parts of the earth. You can see in the case of maize we can have 20 tons per hectare under ideal conditions. In sub-Saharan Africa they're lucky to get a ton. So we're losing 95% of our yield to something, and that something is what we call stress, abiotic or biotic stress. Paddy rice grown in water in Africa is losing 80% of its potential yield. Common bean, which fixes its own nitrogen in Africa, is losing 90% of its potential yield. So there's a lot of things associated with food insecurity. But a very primary principal direct cause is we're not getting good yields because the plants are under stress. You'll notice even in the US here, we're not approaching potential yields. So even in US corn production, drought is the single biggest risk. But in these low input agriculture countries like in Africa, it is a pervasive principal cause of food insecurity. And economic development, because most people are small farmers depending upon their production for their economic livelihood. What's causing these problems? Some of the big ones, of course, drought. The principal risk to crop production worldwide, and it's expected to get much worse in the future. This is a very scary projection that was published a few years ago by the National Center for Atmospheric Research based upon fairly conservative estimates of what's going to happen. And I think in this series, in fact, you have some people coming and talking about climate change. So I won't go into any kind of detail about this. But when I look at these maps, and the purple color here corresponds to the Great Dust Bowl Depression in the 1930s, what we're seeing is by mid-century, the end of irrigated agriculture in North America, in the U.S. anyway, and Western Europe. This is very, very alarming. And trying to sustain 10 billion people under these conditions is inconceivable based upon our current agricultural system. Nitrogen is another big problem. Some places like China are using too much, but most of the world is actually in negative nitrogen balance, which meaning every year they have less nitrogen available than the year before. Phosphorus, a pervasive limitation to life on Earth. Any red color here represents suboptimal phosphorus availability. So these are some of the principal constraints. If you're looking at any particular location, might have a disease or an insect or something. But if you're looking at the whole planet, water, nitrogen, phosphorus explains a lot of crop production. Unfortunately, soils are getting worse. This is a core scale map by the United Nations of soil degradation, mainly erosion, but also salinization. You can see even in the US there's significant degradation, and in Europe. In the developing world, for example, Africa, 75% of cropland is already degraded. So this is an ongoing problem. Every year the soils are worse than the year before. So the outlook is bleak. What can be done about this? Integrated soil fertility management consists of using inputs when you have them. I'll come back to that in a minute. Managing the soil, preventing it from being eroded, and so on and so forth. This is very important. It is inconsequential in the developing world. If you travel around the developing world, you'll see very few examples of good soil management. In fact, my neighbors in Pennsylvania are farmers. They don't manage their soil correctly. And this is the richest country on earth. Since the 1940s, we've had numerous federal programs promoting soil conservation in the U.S. We're not doing it here very well. They're not doing it in Africa. They don't have the technology and know-how. They don't own their own land. They don't have the resources, whatever. This is very important, but it's not a very effective way to address this problem globally. And then the third leg would be using the right crops, or the right genotypes of the right crops. And this is what I'll be focusing on today. But before I do that, let me come back to this issue of access to fertilizers. Because in these low input systems, they just don't have access to fertilizers. This is recent data from the World Bank on global fertilizer use. So we've made this map on a log scale as kilograms of nutrient per hectare of arable land on a national basis. And you'll see that these red colors in Africa correspond to single digits, which is totally insufficient to meet crop needs compared to the hundreds of kilos that we see in the rich nations. And these trends are negative. Fertilizer costs have tripled, quadrupled over the past 10 years. There are places, for example, Malawi, one of the places we work, where fertilizers are significantly more expensive there than here because they do not manufacture them there. They have to import them, and by the time farmers see them, it is well beyond their economic means to pay for them. So even without mentioning issues of is there enough phosphorus on Earth to support global agriculture, can we continually fix nitrogen from the air with the Haber-Bosch process? Even without getting into these kinds of questions, there's simply insufficient fertilizer use in the poor nations to meet crop needs. So for the foreseeable future, this is going to be the most promising avenue. Well, can we do it? That's what we're talking about today. As plant biologists, can we do this? First, I'm going to tell you two stories. The first one's about this plant, the common bean. Most important food legume on Earth. You probably know it as string beans or navy beans, pinto beans. These are all the same species. And there's about 1 billion people on Earth who rely upon this as an important source of nutrients, especially when they don't have animal protein. So this is the typical bean production system in Latin America. Here's a community, here's the church, and here's somebody's home, and next to this home. This is an eroded volcanic landscape that's very low in phosphorus. You can see this is a maize bean system that's very ancient. These bean plants are severely phosphorus deficient. I expect, by the basis of the symptoms I see in this picture, I expect this plant to lose about 90% of its yield. And that's where world average yields are, at only 10% of yield potential. And this is the main reason, it's low phosphorus. So I began this work after finishing my PhD at Davis, finally left California, went to Colombia, South America, the headquarters of the International Center for Tropical Agriculture, where for 20 years, 4 times a year, they were doing massive field trials to try to find bean plants that would beat this guy, a Brazilian landrace called carioca. So it's a low phosphorus field. Their approach was to throw hundreds of materials out there, season after season, trying to find something that would beat this, and they failed. A month before I arrived, they had just fired their soil scientists, reassigned their breeders, and the breeders had written a paper saying it's impossible to improve on carioca, we should give up. I didn't find this very satisfying. I didn't like this approach. There are several things about this approach that I thought would lead to this conclusion. One is they're just looking at yield. The other is they're just looking at a particular location. So for instance, this plant here might have very good phosphorus uptake, but it doesn't have the environmental adaptation to yield well in this location, this photoperiod temperature situation. The other is there's diseases present here. This might be disease sensitive, so we don't see its ability to take up phosphorus. The other thing is this is material that had already been screened for disease tolerance and yield potential, and then as the last stage in the program, the breeders would throw it into this, what they call a purgatory plot, to see what could survive low phosphorus. So they'd already narrowed the germplasm base before they sampled it. I thought, no, no, no. This isn't the way we screen for disease resistance. This isn't the way we screen for insect resistance. Why do we think phosphorus would be any different? Why don't we look just at phosphorus uptake and no other problem? Why don't we look at a lot of germplasm, find the best traits, find the best parents and genes, and then combine them in some scientific way? I thought that's what breeding was. Okay, this is just some kind of brute force thing that wasn't successful. So I convinced, a few breeders to work with me on this, and over some years, to make a long story short, we were able to identify traits that would enhance phosphorus acquisition in this species. And we didn't start off thinking it was going to be roots. We had an open mind. We looked at a lot of different things, but over time we realized, of course, it was the roots. And in fact, it was specific things about the roots, because phosphorus is very immobile. It doesn't move in soil. It stays in the topsoil. So basically, root systems that would concentrate root foraging, topsoil foraging in the topsoil did better than genotypes that were deeper. Let me give you an example. I'm not going to go through and detail all these things, just to give you a few examples. Here's one we noticed early on. Deep versus shallow axial roots. These main root types here are called nodal roots in beans, and these ones are going deep, and these ones are going shallow. You see the genetic variation. These are plants we dug up in the field in Honduras. And of course, this one's exploring the topsoil better than this one. So to show that this is useful for breeding, we used parents, these parents here, crossed them, developed a set of lines that were progeny descending from the same two parents, but that had different root architectures, because it's a quantitative trait. And this is what we see. We put these different recombinant inbred lines, these progeny, in a low phosphorus field, and we look at variation in the trait, and we look at phosphorus uptake. And in this case, we see about a 6-fold variation in phosphorus uptake associated with this trait. Genetically, this is a simple map because this was done some years ago, more than 10 years ago. But so here's the 11 chromosomes of bean, and we did a QTL analysis. And red is performance in low phosphorus fields in the tropics, and the yellow, blue, and green are measures of root shallowness in a plastic bag 9 days after germination in the lab. And we see that there are areas of co-segregation, like here on chromosome 11, where we have shallow rootedness and good performance in the field in the same location, which tells us if breeders select for shallowness, they're also going to get good performance in the field. Here's another trait that is interesting. This is not something we discovered. This has been known for some time, that root hairs are very important for phosphorus acquisition. They go out into the soil and explore the soil better. This is the result of scientific breeding. This is a landrace that has been grown for centuries in low phosphorus fields in highland Peru. And you can see that for some reason or another, the elite lines don't have good root hairs. So once again, we use these as parents. We develop progeny to study how important this trait is. In this case, here's the progeny. Here's the field they're growing in Costa Rica. You can see this is a normal farmer's field. There's nematodes and weeds and different things. It's also low phosphorus. And we're looking at shoot phosphorus content versus our root hair length measured in the field. And you see a very nice correlation for field work. And we can basically increase phosphorus acquisition by a factor of 2.5 by incorporating long root hairs in these plants. So here's an example of two traits we found: root hairs, anatomical trait, and root architecture, root shallowness that help phosphorus uptake. Now for breeding, of course, oh, by the way, this is a relatively simple trait. 6 loci explain over half the variation. For breeding, we're interested in integrated phenotypes. Not just a single trait, but how multiple traits work together. And so we're interested in how these two traits work together. So this is a study we published recently with Magalhães Miguel in Mozambique, where we have recombinant inbred lines that vary for both of these traits I showed you. Both long and short root hairs and deep and shallow root architectures. So the worst combination was short root hairs and deep roots. And in the field, we're just, for this graph, I'm just calling that the baseline. If now we look at a set of rills that from the same parents, but now they have shallow roots, they grow 60% better in this low phosphorus field in Mozambique. If we look at lines that have long root hairs, they grow 90% better. So if we have long root hairs and shallow roots, they should be 90 plus 60. They should grow 150% better, right? No, they actually grew 300% better, which is logical, because root hairs are especially useful if they're in the topsoil because they're on shallow roots. So there's kind of an obvious synergism, we're calling it phene synergism, between these two traits. And I think these kinds of trait interactions are very common with roots and maybe plant traits in general. And we really have to think about that if we're interested in having an impact. How do these things stack up? Another thing we have to think about is trade-offs. Is that something might be good for phosphorus but bad for something else. In this case, shallow roots might be good for phosphorus, but obviously for drought we want deep roots. And farmers in the third world have to deal with both drought and low soil fertility. That's something we have to think about. So here's the study we did in Honduras. Here's the background here where they have both drought and low soil fertility. Here's a set of lines from the same two parents, but now they vary for root shallowness. They grow the same under controlled conditions. Under low phosphorus, the shallow genotypes grow better as we expect. And under drought, the deep genotypes grow better as we expect. So that's a challenge. Breeders don't want to produce a phosphorus-efficient bean line that's drought sensitive. That's a non-starter. So we had to think of some way to come up with a trait that would co-optimize water and phosphorus acquisition. In the case of this species, we did discover such a trait. It turns out these axial roots, which are called basal roots in this species, occur in distinct positions, which we're calling whorls. Here's a genotype that makes 1, here's a genotype that makes 3. Each whorl, because of the anatomy of bean roots, can make 4 roots. So basically, this guy can make 4 basal roots, and this guy can make 12. In fact, if you measure the number of whorls in a lot of genotypes, both wild and cultivated, you see the more whorls you have, the more roots you have. And the idea was, the hope was, that by having more positions, then we can have a broader range of exploration of the soil and capture both shallow and deep resources. That was the hope. Here's what they look like in silico. These are computer models based on empirical data of what these things look like. So this is 1 whorl, 2nd whorl in a different color, 3rd whorl, and 4th whorl. So this is once again field work in Mozambique with Magalhães Miguel. Here's this red soil in the background with low phosphorus. These are lines with the same genetic background, but they have either 2 or 3 whorls. And under low phosphorus, we got about 60% greater growth if we went to 3 whorls. So good. It's good for low phosphorus. How about drought? This is the work of Katie Barlow at our site in South Africa. Katie had a population that varied, same genetic background, but now either 1, 2, or 3 whorls. No yield difference, no growth difference under well-watered conditions, but under drought, we have 70% greater growth. So here's a trait that helps us with both shallow and deep resources. In this species, we have a trait that can do that. This is under very simple genetic control. 2 loci explain well over half the variation. Another way we can do this is instead of telling a breeder to come up with an ideal plant that can do everything, we can have a combination of plants that look the same to the farmer, but they're actually different genotypes, called a multi-line. So we could have a shallow-rooted line good for phosphorus, deep-rooted line that's good for water, put them in the field together, and in fact they have less competition with each other because they look different below ground. And then they're optimal or do better under a variety of stress conditions. Now, this is the idea. This is the work of Amelia Henry, who's now at Erie in the Philippines. So we have, these are closely related, so they look the same. The farmer doesn't have to do anything different with these two lines, but they're actually two different genotypes that we mix together into a multi-line that has both shallow and deep. So Amelia grew these under a range of stressful conditions in Honduras, on farm, on small farmers' conditions, going from very low yield to pretty decent yields. The multi-line yields are in red, and the average of the single-line yields are in blue, and we've got a consistent advantage to the multi-lines across these environments, which means that the growers and the breeders are better off combining two lines than developing one ideal line. So here's another approach to this problem. Another thing we had to think about is our sponsors were very concerned that we were going to be developing plants that mined the soil. They said, okay, you can develop plants that have more phosphorus uptake, but in so doing, are you going to wreck the soil for future generations? Good question. Hadn't been researched. We were hoping it wouldn't be a problem because of this type of concept. Beans are grown on hillsides. Typical bean production system, let's assume 1 ton of yield, twice the world average, just to keep the numbers simple. So 1 ton of yield, we get 2 kilograms of phosphorus in the seed. Every year you're taking out 2 kilograms of phosphorus in the seed. And so if we double yields, we're going to take out 4 kilograms of phosphorus from the seed. We're mining the soil. However, the biggest loss of phosphorus in this system is actually erosion, which can lose significant amounts of phosphorus. And by having bigger plants now, we're losing less phosphorus, so I think we might actually have a net benefit by having the extractive genotype. That was the hope. This again was part of the work of Amelia Henry, her PhD work. Here she is in Costa Rica working with, she went out in the Costa Rican farmland and spent like 1.5 years out there with these people. She rented some of their land, so this is actually a bean field in Costa Rica. You can see how steep it is. She rented it, she installed these, she went to the hardware store and got some tin and invented little lysimeters so she could monitor over 1.5 years the phosphorus runoff versus the phosphorus offtake. You can see the genetic differences. Here's a traditional line, deep-rooted. It's not covering the soil very well. Here's one of these new shallow-rooted lines we had developed. It's covering the soil better. And Amelia found that she could reduce phosphorus runoff 50 to 70% by using this genotype that actually took more phosphorus up. So there was a net benefit for the system by using these extractive genotypes. Not to mention more food, more income, possibly more fertilizer purchases, and these kinds of things. So where's Carioca, that unbeatable bean line? I can't get the breeders to even use it anymore as a bad check because it's so bad compared to the new stuff they've got. Here's Carioca. Here's some of these new lines. This is data from Steve Beebe, head of the bean program at CIA. So here's Carioca, that unbeatable bean line. You fertilize it, you get 2.5 tons, no phosphorus, you get 700 kilos, which is a very realistic yield for the tropics. Now these 3 lines have these shallow roots we've selected, no phosphorus fertilizer. They all yield about the same when they're fertilized. This one yields 3 times as much as Carioca without fertilizer, over 2 tons. These are twice the yield of Carioca. Significant gains by selecting for these root phenotypes. And these are being used as parents in CF breeding programs throughout Africa and Latin America. Following up on this work, several sponsors are helping us push this technology forward in Africa in beans. Here's 3 of our key colleagues. Magalhães Miguel, you've seen him before. Celestina Joshua is a bean breeder from Mozambique. This is her husband, Schwarz Serinda, is the agronomist in the government of Mozambique. So Celestina has been selecting for bean lines with long root hairs, high basal root wall number, and several traits we think are important for phosphorus. She's released lines 2 years ago that were yielding up to 3 times better in regional field trials. So nice impact in terms of more yield. So Schwarz has been showing that these lines have significantly greater nitrogen fixation, that they reduce soil erosion, that they don't bother the maize too much that they're often grown with as maize bean intercrops, and that they're better able to utilize locally available rock phosphate. So we were feeling pretty good about ourselves. We had better bean lines that were improving the environment and more food availability, and our sponsors were not impressed at all. They don't care about that stuff. They care about people. So they insisted that we look at how individual households and villages would be impacted by this new technology. And so we worked with social scientists to do that. This is Jill Findice, now at the University of Missouri. She's a social scientist on this project. So she would do hundreds of interviews with all kinds of people before and after introduction of the new beans and try to track what impact these new lines had on people. before we released the new line, she would do these kinds of pre-hoc impact assessment studies, where it's like, this is carioca. And we would ask people, what would happen if we could increase the yield of this carioca bean? So this particular line, in fact, has no commercial value in Mozambique. It's not sold. It's not bought. It's just grown and eaten, which is important because beans are a major food source for these rural households. So if we ask women what would happen if we increase the yield of carioca, they would say, great, I would have more beans for my kids, which is very important because 70% of the people in Mozambique are food insecure. So that's the kind of outcome we were hoping would happen. Now we have to interview women with female interviewers when the men are not present. And then we interview the men with male interviewers when the women are not present, otherwise they might influence their answers if the other gender is present. So the women were very happy to see that we might improve the yield of carioca. When we ask people about this one, this is the sugar type, this is the main commercial type in Mozambique. Of course, commerce is handled by men. The men would say, great, I can take that extra yield, and what will they do with the extra yield? They will buy beer. Now this particular individual, I don't know how he answered, I'm just showing you a random picture. In hundreds of surveys, and not our project only, but other projects have gone through Africa, what's one of the main things men do with extra money is buy beer. Now what the men will tell you is, this is actually the best thing for the family, because they do not own their own land. And by drinking beer with the village headman, their family might get a better allocation of land in the next cycle. So he's actually being altruistic by drinking beer. At least that's what he says. The women are not so sure. Now this one I have seen, this is an elite class that sells for up to $1.90 a pound in Mozambique. Average wage in Mozambique is 70 cents a day. You guys are probably paying $1 a pound for dry beans. They're paying $1.70 a pound, $1.90 a pound, $2 a pound. This is a high value crop. If you have access to an urban market, you can sell this. You can make a lot of money selling this. So the guys got very excited when they thought that we could increase the yield of these guys. What are they going to do with the extra money? They've already drunk beer with the village headman. Now they're going to set up a new wife. Turns out this is a polygamist area, and the more success you have in farming, the more women you have. So it's just, I'm not going to get into the details of this. It's very interesting, but just to show you, you come in as a plant biologist making much improved yield, and there's all these other impacts you haven't thought about. So Jill has gone through very elegant studies mapping entire villages, every household, how people are related to each other. There's a typical rural household. And she looks at how people share bean seeds. How would this technology move? You know, you don't order beans in the mail there. It's not, you can't buy bean seeds, you get them from your friends. So if we introduce a new bean variety, how does that move? How does that technology disseminate? Well, it turns out the women's and men's networks are quite different. This is just a small, tiny piece of her analysis to kind of show you the kind of things she's seeing. The women's networks tend to be more isolated because they're staying close to home, taking care of kids and beans. They don't move around that much. The men are drinking beer, trading, and all kinds of stuff. They have a bicycle. So they actually can move further. So if we introduce a bean that gets them more beer and more wives, that technology is going to move. Whereas if the women get a new bean that helps them feed their kids, that technology might not move. So there's some kind of unique challenges in trying to move this technology in these low-input systems that Jill's had to deal with. So look, that's the bean story I wanted to tell you. Not only beans, this also worked with soybean in China. This is my dear friend, former student, Yan Xiaolong, who tragically passed away of cancer a few years ago, but he led a team in China to do what we had done in common bean, but with soybean, with funding from the McKnight Foundation. This is what soybean roots look like after several thousand years of cultivation in China. We thought they should look like this. Xiaolong's team selected for shallow roots and had tremendous success in these red, low-phosphorus soils of South China, where soybean is a food security crop. So here's a current cultivar compared to one of these new lines. So this group in South China has now released 15 phosphorus-efficient soybean lines that are growing throughout South China and having significant impact. So this is not just a bean story, it also works for soybeans. Of course, soybeans and beans are pretty similar. What about maize? It turns out these same kind of traits work in maize. Different genes, but same kind of traits, and we can get phosphorus-efficient maize lines. But another problem with maize, of course, is not just phosphorus, but water and nitrogen. It's a huge plant, as you know. It needs a lot of water, a lot of nitrogen. Poor nations, water and nitrogen strongly limit food production. Drought's the biggest risk in rich nations. Nitrogen fertilizer is the primary energy, primary economic, and primary environmental cost of growing corn, and any crop in the rich nations. Cost of nitrogen fertilizer has risen dramatically along with energy costs. Amazingly, over half of applied nitrogen fertilizer is never even taken up. It goes directly into environmental pollution. So this seemed to be like low-hanging fruit that we would like to look at. Now this is the second story I want to tell you. I want to tell you a story about maize and water and nitrogen, and what we can do about that. Just like, remember, phosphorus is shallow and stays in the topsoil. Nitrogen is mobile. This is what happens in Pennsylvania in the spring. This is a pulse of nitrate. This is a very detailed computer simulation, although it looks pretty simplistic. It's actually not. So here's what happens. The rain pushes the nitrate down into the soil. Here's a maize plant growing at the same scale, and you can see it's a race. This pulse is going to be leaving the profile. Some of it's going to be lost before the plants can capture it. So our idiotype, our ideal plant that the breeders would focus on, I'm calling steep, cheap, and deep. A combination of traits that give us deep, rapid exploration of the subsoil to catch that stuff before you lose it and to get that deep water that's down there even in a dry year. Let me just give you a few examples. Steep, meaning like architectural traits. Let's take a look at optimal number of roots, optimal number of axial roots. Huge variation in this trait. We looked at 5,000 maize lines in a diversity panel. And some of them only had 5 axial roots, these crown roots here, very few. And some had, in this case, 35 or 40. Huge variation. These ones over here. And so the question was, do they really need that many? Here's a computer simulation of, these are seminal roots. Here's the nodal roots, the crown roots coming out here. And this one you see has fewer, and this one has more. This is a SimRoot. It's a very accurate model that takes into account the costs and benefits of different traits. And so the idea is the color here shows you how close they are to each other, that for nitrogen capture and also water capture, which move in the soil, that having too many roots close together is a waste of energy. They're competing with each other, whereas having fewer roots is cheaper and you still adequately explore the soil. This was shown in the computer models where we changed the number of nodal roots and we look at growth under low N, and it looks like as we go from 60 down to 20, we get a huge benefit in growth of plants by reducing the number of roots. Kind of counterintuitive. Fewer roots, better resource capture. So we looked at this in a variety of environments. Here's South Africa. We have low and high nitrogen. We also looked at it in the USA in the field and in greenhouse mesocosms. This is work of Pentampong Sangwalai. And so in most cases, this work has just been recently published. I'm not going to go over all the details. If you don't believe me, you can look at the papers. I'm just going to summarize what we found. So in this study, we found that the low crown root number lines had 45% deeper rooting, 200% better capture of labeled nitrate placed deep in the soil, 30 to 90% better nitrogen accumulation, leaf and canopy photosynthesis, 25 to 85% better growth, and 180% more yield. So that trait seemed to be really good. Here's another trait, few long laterals. Now we're talking about lateral roots. Here's the phenotypes we see. Huge variation for these phenotypes in roots. This one has fewer roots that tend to be longer because now each root is competing less for resources from the plant. This one made a lot of lateral roots which tend to be shorter because they're all fighting over the same plant resources. Here's what they look like in the field. This is roots grown up dug up from the field under water stress. So here's what we're calling the many short phenotype, the few long lateral root phenotype. So here we've pulled out one crown root so you can see its laterals. There's many, but they're short, and there's fewer but long. FLMS, same genetic background. So here's what the computer model tells us. Once again, this is a model that takes into account carbon costs and resource capture. It's a physiological model. There's a chute in the model that we just don't see here. So here's the few long, many short, and you can see the model can simulate that pretty well. And the model tells us that the optimal lateral root branching density for nitrogen capture is about 7 under a range of nitrogen regimes. If you have 7 lateral roots per centimeter, you're going to do pretty well. But under phosphorus, that's not enough. The optimum is to shift it. So you need more to explore the soil for phosphorus, which is immobile, than you do to explore the soil for nitrate, which is mobile. That's what the model told us. We never just believe those model results. We always have to validate them. Once again, field work in South Africa and the USA, and greenhouse mesocosms. This is work of John, who just published this recently. So we got significantly less respiration from these plants, deeper rooting, better nitrogen capture, better photosynthesis, better growth, and more yield. So here's some of the growth data in the 3 different environments. So the few long is open bars, many short is closed bars. So you can see under nitrogen stress, these are doing better than these, just like we expected. And Jen also did this under drought. So we did a series of field studies under drought, and in the greenhouse here, so you can see some of the drought effects we're seeing here. So once again, she found cheaper roots, deeper roots, better water capture, deeper water capture as seen by O18 analysis, better plant water content, better stomatal conductance, better leaf CO2 assimilation, better growth, and 61% more yield. So here's lateral root branching density versus plant dry weight in the different environments. As you can see, as you get more density, they do worse under drought. And here's yield. So we can improve yield 61% by selecting for this trait under drought. So that was pretty promising too. Then there's a series of traits that I'm calling cheap, meaning the cost of these roots. How expensive are they for the plant? Let me give you this one example here of high RCA, that's root cortical parenchyma. Here's a laser ablation of a maize root. The green spaces are air spaces created by programmed cell death, hollows out the root, which is stimulated by a variety of stresses. And we showed some years ago that the more parenchyma you have, the cheaper the root is. Obviously, you're turning living cells into airspace, you're going to reduce respiration and also nutrient content of that tissue. There's natural genetic variation for this trait, independent of stress conditions. These are unstressed plants, closely related genotypes. This one does not make parenchyma. This one made huge amounts of parenchyma. So our modeling, once again, this is SimRoot. Now we're looking at pseudocolor image of where the parenchyma is. We know where the parenchyma is. You can see it's a developmental trait that happens in older root tissue. And then the model takes into account how this affects respiration, nutrient content, resource capture, and so on. And the model tells us that plants do better under stress with the parenchyma. So here's exactly the same plants, but with without parenchyma, growing in a low phosphorus soil. And over time, the one with parenchyma does better. Because each unit of root is cheaper, grows a little bit more, catches a little more phosphorus, shoot grows a little more, there's more photosynthesis, there's more carbon. So this is kind of autocatalytic benefit. A small difference in root cost ends up with a large difference in growth. In fact, this study showed that this is the amount, the reduction of the plant by stress, and this is the benefit caused by parenchyma. That under low nitrogen, we could improve, the model said we could improve the growth of plants by almost 60% by having parenchyma. Also for phosphorus, even more benefits for potassium. And even under high yield conditions, we could reduce the nutrient requirement to reach those conditions by having parenchyma. I admit to not really believing these results when we got them because they were so optimistic. So this is Patempong again, doing field work, seeing if it really works. In the greenhouse, in the field, we got much deeper roots with the high parenchyma lines under stress than the low parenchyma lines. In terms of growth, see that's the nitrogen field in South Africa. We had a 60% increase in growth by higher parenchyma lines. In the greenhouse, a 60% increase in growth, just what the model predicted. And in Pennsylvania, where there was slightly less nitrogen stress, we had a 33% increase in growth. And we had a 60% increase in yield under nitrogen stress because of this trait. Now, what we're seeing is anything that's good for nitrogen capture is also good for water capture because it's those deep roots that make a difference. So here we're looking at water stress. Here's these rainout shelters that come over the field when it rains and creates drought conditions. Even in Pennsylvania, which it was shocking to me to find out as a Californian, water falls out of the sky. All through the year. So if you don't have a rainout shelter, you don't get drought. This would be easier in California. But so here we have these stress conditions. This is Jinming Zhu's work. And so Jinming was comparing high and low parenchyma lines, much deeper rooting under stress in the high parenchyma lines, much better plant water status under stress in the high parenchyma lines, and 8 times better yield under stress. This is what the yield looked like. Well watered, the high and low parenchyma lines were the same. The stress lines without a parenchyma look pretty bad. This is looking pretty good, 8 times more yield caused by this trait. So this is being moved into breeding programs. This is some field work in Malawi, working with small farmers on their conditions to do evaluations of Malawian lines. This is work of Joseph Chimungu. So under 14 farmers' fields in 2 regions. The higher income lines, they gave us much better yield. This is over double the yield. On-farm field stations, this is on-farm. The field stations in Malawi, significant increases of higher income lines under water stress, and this is like more than double. This is more than double. Huge effects. So this is very exciting. So now that we know that cheap roots can make a difference for nitrogen and water, we thought of other traits that can give us cheap roots. Here's 2 new ones people hadn't thought about. Changing the number of cortical cell files in the roots. This is more and less, and then changing the cell size. Here's the first one. Changing the number of files. Clearly more files, more expensive. So there's a lot of genetic variation for this. 300% genetic variation in these maize lines. And when we do the field trials and greenhouse studies, we see there's less root respiration, deeper roots, better water capture, greater relative water content, greater stomatal conductance, greater photosynthesis, greater biomass. And here's the yield data. So here's in Pennsylvania and in Malawi. The red bars are yield under water stress. So here the low cortical cell file number lines are yielding significantly better. This is more than double in this case. This is almost double, and this is significant, like 33%. So we're getting nice yield gains from this trait as well. Here's the third trait, which is cell size. Differences in cell size, larger cells, mostly vacuole, cheap. So large variation with this trait, 10-fold variation in maize. And once again, field trials and greenhouse trials, less respiration, the same story as before. Deeper roots, better water capture, better plant water status, better stomatal conductance, etc., better growth. And here's the yield data. We're getting about double the yield by having large cells. This is over double yield from this trait. These are huge effects. So I think this is a promising avenue, is cheaper roots. In fact, there's been a lot of research over the years on water use efficiency in terms of the water we spend to get carbon. We might have been looking at the wrong half of the plant. If we're looking at it from this side, the things I've been telling you today is more this equation. You're spending carbon to get resources. And these traits that we're looking at here today are affecting this part of the equation, and they seem to be really important in terms of breeding prospects. And these are already moving into breeding lines in Malawi. This is Moses Maliru, our colleague, maize breeder in Malawi, releasing and evaluating some of these carbon-efficient root lines in the field in Malawi. Clearly, for breeders, if you say, let's select for root traits, the question is going to be, how are we going to look at root traits? We've got to look at thousands of plants. What do you want us to do? So phenotyping is clearly an obstacle. Here's what we've done. Shovelomics is, all you need is a naive graduate student and a shovel. And no, Jimmy's actually not naive. But so it's about 2 minutes a plot. That's not like, okay, go. That's like all day, how many plots did we do in 8 hours? It's about 2 minutes per plot. And you basically dig up plants and wash them and look at them. We're going through about 25,000 plots a year using this method. So here's South Africa. We dug up a bunch of maize plants. We're scoring them, and then we photograph them. From the photograph, we do this automated image analysis called DIRT. It's uploaded to digital imaging of root traits. It's uploaded to a server at Georgia Tech. They do all the analysis for you, give you the data back, store the image. So here's an example. So here's crown root angle in maize. You see lots of variation. Using this phenotyping platform, here's GWAS. We have candidate genes that are expressed in the roots that are related to hormone action. It makes sense that they're related to angle, identified through this quick and dirty field screening. For anatomical traits, we were originally hand sectioning with the razor blade. It was taking forever. We came up with a new method using a laser called laser ablation tomography. This is a used military laser we got for a 90% discount. And here's a piece of root that we're going to obliterate with this laser. And we're going to photograph it while we're doing that. This is a real-time image of a root in the last moments of his life. As we photograph it, we get a three-dimensional data set that then allows us to do this anatomical analysis. This is a low-resolution quick scan. It took about 30 seconds to get this image. And you can see you can pull out parenchyma and other traits from these samples. In fact, it works even in soil. Here's bulk soil with the root in it. We're just blasting through. And you can see we're looking at mycorrhizas and stuff. And so this is a cool technique. It's very fast, very cheap, very low-tech, run by wage payroll people at minimum wage. So we can do many samples. And then we get those images and run it through a root scan image analysis to pull out our anatomical traits. So just to show you, we can get genetic signals from this kind of means. Here, once again, is a candidate gene that is expressed in the right tissue. It's related to programmed cell death. Makes sense for this trait we're looking at. So I think we have high-throughput phenotyping platforms for these traits. Of greater concern in terms of phenome analysis is that these architectural traits that you see at this scale are going to be interacting with these anatomical traits that you see at this scale. So we have to just remember back to the beginning of my presentation, I talked about hairs of beans and angles and how they interacted. So we have to think about how these things all interact with each other. In fact, here's what the modeling tells us. Here's two of the traits I showed you today, parenchyma in maize and nodal root number, and here's how they perform under low nitrogen. And as they interact with each other and they interact with nitrogen, same for phosphorus, there's a complex response function. The value of a trait depends upon the expression of other traits and the environment. So there's a lot of combinations of interest. In fact, between nitrogen and phosphorus, all the anatomical traits we're working on, all the architectural traits we're working on, there's actually 10 to the 23rd scenarios of interest. That's a lot of possibilities. So Harini Rangarajan in the lab spent 1 million hours of CPU time and a genetic optimization algorithm to go through all 10 to the 23rd scenarios. Well, the way that works is you don't have to go through each one, but you search that space and locate only 250,000 interesting possibilities, which is this graph. And I don't want to dive into the details of this, but just to kind of give you a feeling for the complexity of the phenome. It's one thing to say, what's the utility of this trait, but how it fits into an integrated phenotype in an environment is a non-trivial task. And there's trade-offs. Engineering, they talked about the iron triangle. You can have fast, cheap, or good, but only two. There's always that third one you didn't get, right? And so if we're talking about these root traits, maybe you have rapid growth and good soil exploration, but can you also get cheap? Can you get all three? That would seem to break the iron triangle. Let's give one example. Parenchyma formation, which I've been talking about today. We know that it reduces radial transport of water. We know that it reduces radial transport of nutrients. We're trying to figure out if it affects mycorrhizal colonization, because there's no place for the mycorrhizal to live if there's no cortex. We're trying to find out if it affects the spread of fungal pathogens. We don't know these things. Does it affect the penetration of roots into hard, drying soil? We don't know. So before we tell breeders to go and spend years developing lines, we'd like to understand these things better. And there are no papers about these things. The community, our community, hasn't really tackled these issues of the complexity of the fitness landscape of the phenome. So the challenge of translational research is really, for those of us interested in things like agriculture, have an impact out here. Most of us start at this level, and maybe some of us stay at this level, but I think we really should be explicitly thinking about this level to make progress. And to do that, we have to begin to think about phenes. You might have heard me use the word phenes. I did not invent that word. That word is almost as old as the word gene. Gene is from 1911, phene is from 1925. And it's an element of the phenotype. And we haven't really thought carefully about that. It's like, what is a phene? Some people think you can do genomic selection on yield. I think that is so stupid and naive. Yield is not a phene. It's a combination of, it's the result of everything the plant ever did. You want to get down to something more causative, but then these themselves are governed by processes that then maybe there's some underlying phene like a parenchyma or something that's driving this. We have to get to this level to make sense of the phenome. And, I don't have the answer, but I do know we have to think about this better and develop better conceptual models. Think of all the words we have for the genome. And think of the words we have for the phenome. Trait, which isn't even a scientific word. And I'm saying we should use the word phene. That's one. So we need like a couple hundred more words to round up this model. So let me summarize what I was talking about today. Root architecture is important for resource acquisition. Topsoil is important for phosphorus. Subsoil foraging is important for nitrogen and water. Several of these phenes affect the cost of soil exploration, which is really important. Bean and soybean lines for phosphorus acquisition are being deployed today in Africa, Asia, Latin America. The steep, cheap, and deep videotype is still in the early stages, but it appears very promising. We have high-throughput phenotyping platforms, and we have numerous genetic leads. Let's come back to the green revolution. breeding for yield at low fertility has been successful in the few cases it has been attempted. If you don't remember anything else about what I'm saying today, please remember this one. It is possible to breed plants that do better in stressful environments. Aluminum tolerance is an established technology that's being deployed. Phosphorus is working like I'm talking about. Nitrogen is more complex but is promising. Manganese is working. Very few people working on calcium, potassium, magnesium, but they should be feasible. Recent advances in root biology are making it possible to design a root system for a particular environment. In my opinion, the bottleneck is not more genomics, it's at the phenome scale. And we need more information about the long-term impacts of this technology, but it should be benign, because these things are not going to collapse over time like disease resistance will. A phosphorus-efficient plant will always be phosphorus-efficient. It's not scale positive, meaning it's not only benefiting rich people, and there's a push now, especially in Europe and places where they care about the environment, to get plants that are more efficient. Certainly, the need for these things is only going to grow in years ahead because we have more people, a degrading environment, and reduced input availability. The work I'm talking about is a result of many people. Here's the current configuration of the lab. Thank you very much for your attention.
Okay, so for all of you people that thought that gene networks were complicated, well, we now know that there are other complexities beyond that. Questions? Chris?
Have you tried to combine some of the traits that you found in the maize work and see whether you have a synergistic effect like what you saw with bean?
Yes.
Can you repeat the question?
Yeah, the question was, is there trait synergism in maize? There is. We've even published on it. I just didn't talk about it today. But yes, I think this is common, and certainly in roots, maybe in all plant traits, it's phene synergism. And phene antagonism can happen also.
Renee?
I was wondering about the biomass. So when the root have bigger cells or empty space, that's reduced biomass of the root, right?
Repeat.
Or
The question is about the dry weight of the root, because it's more water or something.
or whatever, how you measure biomass.
Well, what we see in every case, whenever we reduce the metabolic cost of a unit of root tissue, the plant makes more root tissue. So there seems to be an allometric control that we don't understand of plants wanting to balance root and shoot allocation. And if we reduce the cost of a unit of root, the plant makes more roots, which is what we're hoping.
So the biomass doesn't go down.
No, biomass doesn't go up. We just have more longer roots now, getting more resources, which then increases overall growth. And if we look under high inputs, I don't know if you noticed, I always have the high input control here. These traits have no effect on plants at high inputs, which is perhaps why we don't see them in elite germplasm. Breeders have been selecting under good conditions. They're not useful under good conditions. They're useful under stress conditions.
Russ?
So mycorrhizae is supposed to be important in foraging for phosphorus. Did you take these sorts of experiments into consideration when doing your work with phosphorus?
Yes, most of our work is in the field where there are mycorrhizas, and we ignore them. We've done experiments showing we can ignore them, so we do ignore them, and it doesn't seem to affect anything. So I think they're important, but they're important for all genotypes equally well. So the ranking of genotypes doesn't, we know they're there, but we just ignore them, and that doesn't seem to affect anything. Now, in the case of Arenchyma, we're concerned that by hollowing out the cortex, there's no place for them to live, and then, so we have a student working on that. But I just think of it as part of the background, There's a lot of cool research on this, and it doesn't seem to be affecting things in the field.
Well, you could read fitness of the mycorrhizal organism to the root, right?
You could, but mycorrhizas are very promiscuous, and they're the most abundant soil fungi, and so it's a hard thing to manipulate. So we're working with breeders, we're trying to go for the low-hanging fruit. So they're important, and they're cool, but we just, in our own work, we just don't need to think about them. And we work in the field, so they're there. And we work in the greenhouse, they're not there, it doesn't change anything.
So one more question, I guess. Siobhan?
So the question is, so mechanistically, do you think that the Arenchyma are performing any sort of function other than just adjusting the rate of biomass and perhaps increasing growth? Like, do you think there's any process by which they're aiding in utilization of the mineral nutrients?
Well, I think,
Repeat the question.
yeah, the question is, what's the function of Arenchyma?
Yeah.
And of course, there's a huge literature on their assistance in providing oxygen for root respiration under hypoxia. They can help the gases, ethylene and CO2, transported in the root. Our work would suggest that there's significant benefits under nutrient and water limitation, which are pervasive on Earth. So I think that they have a large function in making roots less costly. But apart from our lab, there hasn't been a lot of people looking at this, so I hesitate to say that's a given, that's a fact. All plants make parenchyma. The earliest angiosperms are erenchymatous. So, you know, I think we can come back and look at that and say, my question is, if parenchyma is so good, how come all plants don't make it all the time? So that's what I want to know before I tell the breeders to select for it, is like, there must be a catch, you know, and what is that? What is that downside?
And then, is there a relationship between the number of cortex layers and the number of parenchyma?
Well, yeah, things like Arabidopsis, which hardly have any cortex, cannot make parenchyma. But most real plants with big cortexes. I mean, that's the case where Arabidopsis is not a good model, right? A bigger plant will always make parenchyma if you let it. Some plants make these beautiful erenchymatous structures. So it is pervasive in the plant.
Okay, well, let's
Thank you very much.
Jonathan for